I welcome you all. I am Dr. Achal Tandon and we'll be continuing further the part of Robbins of Neoplasia where we left. So genetic predisposition and interaction between environmental and inherited factors. So we all know that the important factors are some of people have germline mutations and some have don't have any germline mutation. So when they are exposed to environmental factors, they develop new mutations. So it is seen that up to 95% of all the malignancies are sporadic. And it is seen that only 5% are germline. So there will be more to it. Like breast cancer risk in females who inherited mutations from parents, that will be BRCA1, BRCA2, tumor suppressor genes have been observed to have up to threefold higher risk of breast cancer. So it is true that up to 95% are sporadic, but these 5% that are having these germline mutations have multifold increased risk of developing malignancies. There are multiple inherited genes, and one of which it has been described, this BRCA and BRCA2. Along a lot of pleomorphisms in enzymes have been seen, like that in cytochrome P450. So this is a cytochrome that is required to convert the procarcinogens into carcinogens. Okay, so the people who are having polymorphism in P450 genes have been seen to have increased susceptibility to smoking associated induced lung cancers. Now we come to very important topic, that is molecular basis of cancer and role of genetics and epigenetics in alterations. Okay, so there are few things that are common to all malignancies. That is what actually happens that leads to development of malignancy. So there is always a first thing that will happen in anybody's body is a non-lethal genetic damage, okay, that lies in the heart of cancerogenesis. That means there was damage to a cell that did not lead to death of that cell, but rather it continued to live with that mutation. So that is in the heart of cancerogenesis. If it would have died, there would not have been any cancer. So it survived the mutation, but it continued to live with that mutation. So this is a diagram showing there was a normal cell that underwent some mutation due to environmental factor that led to initiated precursor cell, but this cell is not a malignant. This is having a mutation, but currently it has not developed a malignancy. When it will develop malignancy? When multiple driver mutations will drive this into causing its multiple high rate of proliferation, doing abnormal repair, inhibiting apoptosis. Multiple mechanisms will be shut down and further due to these driver mutations, and that will lead to founding cancer cells. This cancer cell will also not stop to mutate because it is having such rapid division. Now the cancer cell will give subpopulations. They will all be clones, but these are clones. Each cell is dividing and each cell is acquiring new mutations and developing new phenotypes. Okay, so this will also be genetically heterogeneous. So early in the malignancy, it will have similar it will have similar genetic material, but later in the malignancy it will start to acquire new mutation, and every cancer cell can have different phenotypes. The next point that it writes is the tumor cells forms clonal expansion of a single precursor that has incurred genetic damage. So it says the damaged precursor cells will continue to divide. That is the second thing in the heart of cancerogenesis. One, it has survived, it has not died, non-lethal damage. Second, it is proliferating. Then there are 4 classes of mutations that can occur that leads to malignancy. The first one is proto-oncogene, the second one is tumor suppressor gene, the third one is apoptotic gene, and the fourth one is DNA repair gene. So what happens if these are all normal genes in our body. The proto-oncogenes are bringing signals to divide. Normally, whenever there is damage, brings signals to divide and proliferate that are normal. But what if its signals increase? So if the proto-oncogene signals increase, it can lead to cancer. And what about tumor suppressor genes? Whenever these signals are coming, to stop these signals and to tell the cell that no more proliferation is required, a tumor cell suppressor gene is there. So whenever this cell will reduce its function, it will lead to malignancy. Then apoptotic gene. Whenever there is damage to a cell or it has lived its life and now with multiple proliferations, it has aged. Now it undergoes a normal apoptosis. But if apoptosis function is reduced and the cells are not able to die even in spite of having lethal damage, they survive, then this leads to malignancy. So reduced function of apoptotic genes leads to malignancy. And DNA repair gene. If there is reduced function of repair, there will be increased mutations that will not be able to correct itself, and in the absence of DNA repair, it will not be able to cause apoptosis in the cell. So this will further lead to malignancy. So reduced function of this will also lead to malignancy. So all the three, tumor suppressor, apoptotic and DNA repair gene, reduced function, loss of function will lead to malignancy, whereas proto-oncogenes, increased function, overexpression, amplifications will lead to malignancies. Apart from these, point mutations and translocations can also lead to malignancies. So when the malignant cell multiplies so many types and it is so much dividing, it has increased chance of developing more mutations. So when there is increased chance of developing more mutations, this type of cell has been referred to as the mutator phenotypes because it is marked by genomic instability. Then what happens? The carcinogenesis results from accumulation of complementary mutations in a stepwise fashion over time. So, malignant neoplasms with several phenotypes attributably referred to as cancer hallmarks. So, there are multiple mutations. So, that functions that we have seen, that this is on the growth signals are coming and it is not requiring the growth signals, it is not responding to the inhibitory signals, there is evasion of apoptosis and multiple things. All these hallmarks are required with multiple driver mutations in the malignancy that will be acquired after the initiator mutation. Multiple mutations that are happening will help the cell to acquire all these properties. Then the mutations that contribute to acquisition of cell hallmarks are all known as driver mutations. So there will be other mutations also that are not leading to anything, that are not leading to any change in phenotype because they are useless. There are mutations that are not leading to the eventual malignancy. Those mutations will be known as the passenger mutations and they will not lead to change in any phenotype. But the mutations that are leading to acquiring of the hallmarks are called the driver mutations. So some of these driver mutations will lead the cancer cell to further finally develop cancer stem cell-like properties. Then that I have discussed about the messenger mutations that are not leading to any phenotypic consequence are known as passenger mutations. Mutations in many other genes contribute to tumorigenesis by interfering in host immune response. So normally the CD8 cells will not let any tumor cells to grow in the body, but when the CD8 immune response is weakened or overall immune response is weakened, then these malignancies develop. But some cancer cells acquire some properties that they will prevent from its tumor cells from getting captured by the immune response. This include reduce antigen expression over the cells. This will include hiding of the antigens by the carbohydrate that will be discussed further. Once the tumor has developed, it further doesn't stop to divide. It continues to divide and acquire new genetic mutations. So this has been seen and associated with Darwinian selection, that during the studies of evolution that we have studied, we have seen that only the fittest survived. And this was seen that how one species had developed to another species, there has been some mutations, it developed into another species, there were multiple mutations, multiple species, but only few survived according to the geographical conditions and environmental conditions, the rest all died. So similar things happen in the tumors. Normally, whenever the tumor is detected, it's almost 1 gram, or it is up to 10 to the power 9 cells that has been developed only after doubling of up to 30 times cell doubling. This is the cancer that we will be able to detect. But if you are having a cancer that you are able to detect, that means a lot more times of this cancer has already died. A lot of it has already been apoptosed, because only the best suited has survived, that has fought against all immune response, that has fought against all your body mechanisms, repair mechanism, apoptotic, everything that is a fight through every mechanism that prevents your body from developing a cancer. So this is how it is related to evolution, and further we will see. Then this process that we have seen, that tumor becomes with every, even in spite of being a malignancy already, it will continue to have mother mutation and it will continue to become more aggressive. So a study was done, a study was done in renal cell carcinoma patients. It was seen, the tumor was seen taken from the primary side as well as it was taken from the metastatic side, and all the cancer cells were evaluated and the phenotypes were evaluated, and it was seen that the mutations that were in the primary malignancy were very different from that of the metastatic. So with the time, with new mutations, after it is metastasizing, or the cells that were metastasized also have some more mutations, only they were able to metastasize. Then after metastasis also they were acquiring new mutations to suit to that situation best. So it continues to mutate, and this was correlated with the Darwin's model that it gave during the evolution for the survival of the fittest. So this diagram is the one from the Darwin's that tells that there was one species that continued to live a long time, developed a mutation, developed another mutation, gave rise to 3 species, one of which did not survive, one of which further gave rise to 3 more, further 2 more, further 2 more, and that led to evolution, and only the ones suited to the environment survived and rest did not survive. So this is how he explains the evolution, and similar way is the malignancy, where a normal cells develops an initiating mutation, develops a malignancy. This malignancy further develops a mutation, give rise to R6 population, develops a potential mutation that is compatible with metastasis. So it is giving a metastatic tumor. This is further giving a mutation that is further compatible with another type of place to mutate. Then it is developing in that way. Then it is also saying that not only the mutations are leading to this, but also epigenetic changes. That is, there are some things like DNA methylation, and then there are histone proteins. So in spite of not having mutations, sometimes these DNA methylation, these epigenetic changes can silence the gene expression. So these can also lead to malignancies, as well as these are being used for management of these patients by suppressing these things. So the studies are being going on for using of these properties for the treatment of the patient. Cellular and molecular hallmark of the malignancy. So what are the hallmarks that we were talking about, that it is acquiring, what the cells are acquiring, that is leading, that are the driver mutations that are leading to acquiring of these hallmarks. So what these hallmarks are? These hallmarks are the self-sufficiency in the growth signal, that cancer cell will not need continuous growth factors to divide. It will not it will not be suppressed or inhibited by the growth inhibitory signals like tumor suppressor genes. Then there is altered cellular metabolism, like the normal metabolism will not be enough to feed the tumor cells and make it survive. So it switches to Warburg's effect, that is aerobic glycolysis or Warburg's effect, that helps it synthesize the macromolecules and the organelles. This is the reason why we are able to detect the tumor cells in PET scan. Then we come to evasion of apoptosis. So now these tumor cells will not normally die with normal apoptosis. Then limitless replication potential. So we know that every cell has a limited capacity. Then it will divide up to 30 times, then it will die, or it will divide up to 100 times, then it will die. But limitless replicative potential has been acquired by a malignant cell. How is it? With the help of telomerase or multiple mechanisms that normally we have seen the after multiple replications, the ends of the chromosome blunt and start to get shortened. So these telomerase gets activated and prevents its shortening, and that gives us limitless replicating potential. Then we come to sustained angiogenesis. So when the tumor size is increasing, it will need lot of blood supplied so that it can survive. So the tumor cells cause hypoxia. This hypoxia induces VEGF and leads to angiogenesis that promotes the cancer cells to develop and survive. Then the ability to invade and metastasize. So these tumor cells have developed properties that leads to release of collagenase, that leads to subpopulation will go through blood, will then metastasize and lay its seedings in other organs, and this will also be multifactorial, and these properties are acquired by multiple driver mutations. Then the ability to evade the normal host immune response. The acquisition of genetic and epigenetic alterations that confer these hallmarks may be accelerated by genomic instability and cancer-promoting inflammation. Now we come to self-sufficiency in growth signal or oncogenesis. Oncogenes, what are oncogenes that we have seen? Normally, for a cell to grow, a proto-oncogene is required. That will be a growth factor that will bind to a growth factor receptor for the growth of the cell. Now, if the proto-oncogene functions increases and is not in control, it will be known as a oncogene. So now we will see a diagram to understand, and this diagram will be the basis of complete proto-oncogenes. So what happens is, whenever normally in our cell, whenever there is a signal for synthesis of something, there are multiple mechanisms in the cell that are leading to that signaling. So it has been described here in this. The receptor tyrosine kinase pathway is there, G protein coupled receptor pathway, JAK-STAC pathway, then WANT pathway, Notch pathway, Hedgehog pathway, TGF beta SMAT pathway, and NF kappa beta pathway are there. Of these, the most common one is discussed here. The rest will be discussed in another chapter. The most common and the one associated with malignancy is being discussed here. So what happens is, a growth factor comes here. It binds to the growth factor receptor. This growth factor receptor is located in the lipid bilayer. This is the outer membrane of a cell. This receptor is connected to a bridging protein that is further connected to an inactivated RAS, that is a signal transducing protein. It is inactive because it is bound to a GDP. But when the growth factor will bind here, it will cause the bridging protein to phosphorylate this GDP into GTP. This leads to activation of the RAS. Now this RAS will activate RAF family, that includes the BRAF and phosphoinositol kinase, that will activate AKT and mTOR pathway, and BRAF that will activate the MAP kinase pathway. This will lead to increase transcription proteins, that is MYC, cyclin D, D-cyclin. This leads to cell cycle progression. As we can see, this is the first cyclin This along with CDK4 leads to progression of cell cycle at the first checkpoint, that is G1S checkpoint. Then the MYC protein, that is the most dangerous oncoprotein, and that leads to pro-growth metabolism and increased protein synthesis. The various mechanisms by which MYC leads to malignancy have been discussed ahead. So this is the normal mechanism by how normal cells grow. But whenever there will be increase in function mutation or any component of these will lead to malignancy. Now this RAS, how it stops, how its signaling stops. Once it is activated, then will it lead to endless proliferation? No. There is another protein known as GTPase. This will, as soon as it becomes activated, this GTPase activity starts and augments this function of activated GTPase by 1010 times and again converts this GTP to GDP and makes it inactive so that it works in a controlled manner. So any increase in, as well as there is another inhibited protein, that is a P10 protein, that is inhibitor of phosphoinositol 3 kinase. So anything that will leads to increased function of growth factor receptor, signal transduction proteins, that is RAS, then the RAF, BRAF, phosphoinositol 3 kinase, MYC or cyclin D will lead to malignancy, as well as a reduced function of GTPase activity or P10 activity will also lead to malignancy. So now we will see this in further details. The oncoproteins have multiple roles, but virtue all encode constitutively active oncoproteins that participate in signal pathway that derives the proliferation of cell, that we have already discussed. So we will see what are the growth factors, what are the growth factor receptors. So it is very simple now. You can see there is a growth factor, there is a growth factor receptor, then there is signal transduction protein, the RAS that we have seen. Then there is nuclear regulatory proteins, the MYC, and then cell cycle regulators, that is CDK4 and cyclin D1. So you can see amongst the growth factors there is platelet derived growth factor, fibroblast growth factor, transforming growth factor, and hepatocyte growth factor. The platelet derived growth factors overexpression. So mostly the increased malignancy or increase in function is seen in all the oncoproteins. So growth factor increased function will be either by overexpression or by amplification. Increase in receptor function is either by amplification, by point mutation, or by translocations. And similarly, in the signal transduction proteins also there is point mutations. It is important to note that mostly the translocations that have been described are in association with blood malignancies. Like we can see PDGF receptor translocations in leukemia. We can see that there is non-receptor tyrosine kinase, that is ABL translocation in CML and ALL, and not signaling pathway translocation seen in leukemias, lymphomas, JAK-STAT signaling translocations and point mutations that are seen in myeloproliferative neoplasms and ALL. Rest are point mutations. Then we come to nuclear receptor proteins, that is translocations. Translocation again associated with lymphoma, Burkitt's lymphoma, MYC, and NMYC amplification with neuroblastoma, solid malignancy. Then cyclin D, also translocation associated with mantle cell lymphoma, multiple myeloma, and amplifications associated with the solid malignancies. Now we come one by one. The PDGF is associated with astrocytoma, fibroblast growth factor associated with osteosarcoma, that is HST1. Then fibroblast growth factor 3 amplifications associated with stomach cancer, bladder cancer, breast cancer, as well as melanoma. Then transforming growth factor overexpression also associated with astrocytoma again. So two are associated with only astrocytoma, that are PDGF beta and transforming growth factor alpha, and the fibroblast growth factor with osteosarcoma HST1, FGF3 with 4 malignancies. This is breast, bladder, 2B, stomach, and melanoma. Then hepatocyte growth factor with that can be very well predicted with hepatocellular carcinoma as well as with thyroid cancer. Then growth factor receptor. Then EGF family that we already know. EGF1 associated with adenocarcinoma of lung. ERBB2 that is associated with breast carcinoma, that is a family, her family that we have already studied. Then FMS like tyrosine kinase 3, FLT3. This point mutation is associated with leukemia. Then receptor for neurotropic factors, RET mutation is seen in men syndromes. Then PDGF receptor amplification is seen in gliomas. Then PDGF, we can remember the growth factor is associated with astrocytoma and its receptor is associated with glioma. It's in its amplification, whereas its translocation is associated with leukemia. The KIT ligand mutation, point mutation, is associated with multiple things that are, it is associated with GIST, it is associated with seminomas, it is associated with leukemias. Then ELK, translocation is associated with adenocarcinoma of lung and its point mutation is associated with neuroblastoma. Then we come to GTP binding proteins. Now, amongst GTP binding proteins, are RAS or multiple RAS. So important to here is that KRAS, we can don't correlate the K with the kidney. All the kidney and bladder is associated with the HRAS, and the KRAS is associated with the different colon C, that colon, lung and pancreas. Then NRAS with the name N, it is associated with melanoma and hematological malignancy. The GNAC is also associated with uveal melanomas. GNAS is associated with pituitary adenoma and other endocrine tumors. Non-receptor tyrosine kinase, that is ABL, is associated with BCR-ABL, everybody knows, with CML and ALL, rarely ALL. Then RAS signaling, RAF is associated with melanoma, hairy cell leukemias, it is associated with colon cancers. The Notch is associated with leukemias, lymphomas, especially the T cell type, and it is associated with breast malignancies. JAK-STAT signaling associated myeloproliferative neoplasms, it is associated with polycythemia vera, essential thrombocytosis, also it is associated rarely with ALL. Then nuclear receptor proteins, that is MYC and MYC, and MYC is associated, it is the worst kind of protein that can lead to malignancy, and MYC translocation is seen in Burkitt's lymphoma, and its N-MYC amplification is seen in neuroblastoma. The sign cycle regulators, this is cyclin D1, its translocation is associated with mantle cell lymphoma and multiple myeloma, and amplification associated with breast and esophageal cancer. Cyclin dependent kinase 4, amplification is associated with, amplification or point mutation associated with glioblastoma, melanoma and sarcomas. Now we can see a little description of the things that we have already studied, like ERBB1, that is usually associated with, that is our growth hormone receptor that we can see here. ERBB receptor 1, this is associated with adenocarcinoma of the lung. And then we come to ERBB2, HER2, that is very common and we know that is associated with breast carcinoma. Then we come to ELK. ELK, either it can be translocation. The translocation in the ELK that is seen, seen in a subset of adenocarcinomas, a deletion of chromosome 5 fuses with a part of ELK gene with a part of another gene, that is EML4. This EML4 ELK fusion is seen and it causes increased tyrosine kinase activity. So when we know that there are so much fusions, we know all these mutations and their direct association with malignancies, a question can arise, then why we don't give a direct treatment for these therapy and then final results can be there, like the patient's cancer is cured. But things are not happening in that way, because this particular thing is known as addiction. So if a tumor with a particular mutation is given a direct treatment to a particular mutation and it gets completely corrected, so that is known as addiction. That property is called as addiction. But the tumor, as it requires multiple mutation and it is not the single mutation, then that's why it doesn't respond directly, although some and high level responses have been seen to occur, but still it is very limited. Downstream component of receptor tyrosine kinase signaling pathway. So as we have seen, once the RAS is there, RAS further activates BRAF and phosphoinositol 3 kinase that further activates the MAP kinase pathway as well as the AKT pathway. So this is all that I have described. The one thing that is important is the RAS gene have 3 types. This is HRAS, KRAS and NRAS that we have already seen. The HRAS associated with kidney and bladder, the KRAS associated with colon, lung and pancreas, the NRAS that is associated with your melanomas are discovered. It is seen up to 15 to 20% of all the malignancies are having RAS mutations, and up to 90% in pancreatic malignancies, and in up to 50% in colon, endometrial, thyroid malignancies, and 30% in lung malignancies and myeloid leukemias. Then we can see that I have already described the GTPase that causes conversion, that augments the conversion of GTP back to GDP and reduces the function of RAS is very much required. So any reduced function mutation in GTPase gaps, the GTPase activating proteins lead to malignancy. So it is one thing that will be important to describe is then familial neurofibromatosis type 1. This is the protein that encodes for GTPase activating protein. So if there is this leads to reduced activity of GTPase activating protein, that leads to increased RAS function and that leads to neurofibromatosis type 1. Then there is if there is direct mutation in BRAF, the ones that I seen that RAS activates the RAF. So BRAF is one of the member of RAF family. So when there is mutation in BRAF, that leads to up to 100% it is associated with hairy cell leukemia. In 60% of melanomas it is associated, and it is also associated in numerous colon cancers. Like activating RAS mutations, RAF stimulates downstream kinase and ultimately activates transcription factor. So along the line in this sequence, all the factors that were happening after BRAF sequence will happen in this mutation. Then the second mutations that will be directly affecting the phosphoinositol 3 kinase, the gain of function in phosphoinositol 3 kinase, or we have seen there was a mutation called P10 of phosphoinositol 3 kinase that reduced function of P10. Both of these situations will lead to increase of the pathway that was along and after the phosphoinositol 3 kinase. The phosphoinositol 3 kinase is recruited to plasma membrane associated protein complex. Here, like BRAF, it activates a cascade of serine threonine kinases including AKT. So this AKT phosphorylates multiple proteins, up to 150 proteins it phosphorylates. These proteins include the mTOR, the apoptotic proteins that are bad, FOXO, transcription factors, MD, M2 and IAP. So you can always say, then why don't we make an direct drug that is targeting the RAS? But we have been unfortunately failing in making those drugs. But there have been direct drugs that are targeting BRAF that has given good response in the treatment of melanomas. So this property, as I have already told, is phenomena is called oncogene addiction. Then we come to non-receptor tyrosine kinase. Now we have seen all the mechanism that was happening was when the growth factor was binding to the growth factor receptor. But these are the tyrosine kinase that don't have a receptor on their membrane, but rather everything is regulated through the cytoplasm and the nucleus. So the example of these non-receptor tyrosine kinase include the BCR-ABL, that is very important and seen in CML. Despite its non-membranous location, these oncoproteins seems to activate the same signaling pathway as receptor tyrosine kinase. An important example of this is BCR-ABL. As we have seen, normally there is ABL that is located on chromosome number 9, that is translocated to chromosome number 22 where there was BCR. So now there is a BCR-ABL protein, chimeric protein is formed that leads to increase, because it leads to increased tyrosine kinase activity and increased proliferation of myeloid lineage is seen. This oncogenic mutation is oncogenic addiction is being used, its property and treatment directly targeted towards BCR-ABL has been given using the property of the oncogenic addiction, and it has been seen to reduce a very significant amount, because it is very much important to give the treatment and stop this proliferation, because if there will be more proliferation even in the myeloid series, with the time, with increased proliferation, there is always an increased risk of developing new mutations, additional mutations. And because of those additional mutations, if these therapies are not given on time, the incidence or the rate at which it will develop into acute leukemia will be very rapid. Will be not very rapid, but will be rapid as compared to the other type. So now if we give the treatment, it is not a cure. It will not completely completely stop this this mutation. It will persist in the body. So it will be required to give for the lifelong time. Then there is other, that is the JAK-STAT mutation. So JAK-STAT signaling is another example where there is not translocation, but rather there is increased signaling. An example, the type of mutation that is found in non-receptor. It is a non-receptor tyrosine kinase similar to the BCR-ABL, and it participates in JAK-STAT signaling pathway, which induces mitotic signals for growth factors and cytokine receptors that lack tyrosine kinase activity. JAK-STAT activation alters the expression of target genes that binds to the STAT transcription factor. Several myeloid neoplasms have been seen with association with this. So now JAK2 inhibitors have also been developed so that we can prevent this. So the examples of the JAK2 mutations are, as you all know, in polycythemia vera and essential thrombocytosis. Then we come to transcription factors. This is one of the very important topic. The transcription factors are we know they are MYC, MYB, Jun, FOS, REL. So for it is for MBBS students enough to know about the MYC, but for postgraduates we can know about all these 5. And the MYC is being the most common affected in cancer. So what is a MYC? MYC is a proto-oncogene, is virtually in it is expressed in all eukaryotic cells. So we are seeing that there are different mechanisms. I said there are 8 mechanisms that are further leading to growth, but it is saying that this is present in all. So in spite of different mechanisms that all lead to a single last sequence, that is activation of MYC. So this MYC is very important for oncogenesis. If there is a mutation in MYC, it leads to multiple things, like the MYC mutations like D-cyclins are directly involved in cell cycle progression. So MYC target gene, that is cyclin D, is directly involved in cell cycle progression. Then it upregulates the rRNA and rRNA processing and cause assembly of the ribosome. So this is first thing, that it is directly, its target gene is D-cyclin. Second thing is it is increasing the rRNA and rRNA processing and causing the assembly of the ribosome. Third thing is it upregulates the Warburg's effect, or also it increases multiple glycolytic enzymes that leads to glutamine metabolism and leads to development of intermediates that leads to formation of macromolecules, DNA, proteins, lipids required for the malignant cells. Then it is based on protein effect. This is important, protein effect. MYC can be considered as a master transcription regulator of the cell growth, because at the end of the day all the pathways are finally converging at the MYC. So if there is a mutation or translocation in MYC, it leads to the fastest growing human tumor, the worst human tumor, that is the Burkitt's lymphoma, that shows chromosomal translocation in MYC. And then there is MYC upregulation can also cause, in some studies it has been seen in some studies, that it causes expression of telomerase, that I have already discussed, cause limitless replication potential. Then there is MYC is one of the handful transcription factor that can act together to reprogram the somatic cell into pluripotent stem cell, that it leads to stem cell like properties. Then given the importance of MYC cell regulation growth, so now it is telling that in Burkitt's lymphoma, a subset of B and T cell tumor, the MYC gene is translocated into the antigen receptor gene. So there's a diagram that it is showing, it is explaining the same thing. So there is a chromosome number 8 where the MYC gene is located, and then there is chromosome number 14 where immunoglobulin heavy chain gene is located. So these heavy chain regions are very rapidly dividing areas because they have to they are required to divide very rapidly in order to make antibodies, that is completely made by permutation and combinations. So it needs very rapid divisions. When the MYC gene is translocated and it goes to the near the heavy chain, it also starts to divide very rapidly. So this very dangerous MYC gene, when divides very rapidly, leads to a very dangerous tumor and very fast growing tumor, that is the Burkitt's lymphoma. Then there are other solid malignancies that has NMYC, LMYC. So NMYC is associated with neuroblastomas, whereas LMYC is associated with small cell cancer of the lung. The NMYC, how is it evaluated and how we find out? It can either express as homogeneous staining region. So we can see this homogeneously staining, and or it can be seen as double minutes. Then cyclin and cyclin dependent kinases as mentioned. So I would like you to refer to page number 27 of Robbins to understand this.